What makes pancreatic cancer immunotherapy difficult? Imagine trying to get a message through to a fortress that's heavily guarded and surrounded by thick walls. That's similar to what happens with pancreatic cancer when doctors try to use immunotherapy. Pancreatic ductal adenocarcinoma, the most common type, has a very tough environment that makes it hard for the immune system to do its job. One big challenge is the tumor microenvironment, which is like a dense jungle of cells and fibers. It includes immune-suppressing cells like regulatory T cells and macrophages, as well as a fibrous extracellular matrix that acts like a physical barrier. This environment not only blocks immune cells from reaching the cancer but also prevents them from activating properly. It's like trying to send soldiers into a fortress that's surrounded by walls and traps. Another issue is that pancreatic tumors tend to have a low number of mutations. This means they don't produce many neoantigens, which are the markers that help the immune system recognize and attack cancer cells. Without enough of these markers, immune therapies that relying on boosting the immune response, like checkpoint inhibitors, often don't work well. The cancer's genetic makeup also plays a role. Mutations such as those in the KRAS gene help the tumor evade immune detection from early on. These genetic changes can interfere with the immune system's ability to recognize and attack the tumor, making single therapies less effective. The physical structure of the tumor adds another layer of difficulty. The dense fibrous tissue, called desmoplasia, acts like a shield that keeps immune cells out and prevents drugs from reaching the cancer. This stromal barrier is a major reason why treatments that work in other cancers often don't have the same success in pancreatic cancer. Clinical trials have shown that immune checkpoint therapies, which have been effective in cancers like melanoma or lung cancer, have limited success here. Combining chemotherapy with immunotherapy or using multiple checkpoint inhibitors has not yet produced significant improvements, indicating that we need new strategies to overcome these barriers. Adding to the complexity is the fact that pancreatic cancer is very diverse among patients. Different genetic and molecular features mean that what works for one person might not work for another. This heterogeneity makes it difficult to develop a one-size-fits-all immunotherapy approach. Researchers are exploring personalized treatments based on specific tumor characteristics, but this is still in the early stages. Furthermore, only a small subset of patients with certain genetic features, like high microsatellite instability, respond well to immunotherapy. Most patients lack these biomarkers, which limits the broad use of current immunotherapy options. In summary, the challenges of using immunotherapy for pancreatic cancer come from its highly suppressive environment, low mutation rate, genetic factors that help it evade immune detection, and physical barriers created by dense stroma. While ongoing research is promising, it's clear that overcoming these hurdles will require combination therapies that can modify the tumor environment, boost immune cell activity, and be tailored to each patient's tumor profile. It's important for patients to understand that immunotherapy is not yet a standard, widely effective treatment for pancreatic cancer. Always consult healthcare professionals for personalized advice and stay informed about the latest advances in this complex field.